Hi, we're Group 1, and our names are Karishma,
Madeline.
And Vanessa.
and we'll be diving into how NASA's Perseverance rover searched for evidence of past life on Mars. So, first, for some background information, NASA's Perseverance rover sampled rocks in the Jezero crater to help uncover signs of past life on Mars. And the Perseverance rover landed on Mars on February 18th, 2021. The rocks that it analyzed in a study to prove that the iron phosphate and iron sulfide minerals found in samples were products of redox reaction was part of the purpose of a study to show that there was past life on Mars. Outcrops were explored during this mission, and they were collectively referred to as the Bright Angel Formation. And below is the Perseverance rover on Mars's surface. For the first key finding, we focused on redox-driven mineral formation, where iron phosphate and iron sulfide minerals in the Bright Angel Formation likely formed through redox reactions under non-oxidizing conditions. This supports the idea that iron and sulfur cycling played a major role in how the rock's chemical composition changed over time. And here is an image below of the different textures that were observed in the Bright Angel Formation. For our second key finding, we dove into environmental signs of past life, where variations in color, grain size, and hydration suggest that Jezero crater once experienced fluctuating redox and water conditions. This likely led to the creation of environments potentially favorable to microbial life. And here's a color-coded image of the Jezero crater reflecting changes in rock chemistry, hydration, and oxidation state. Next, for our third key finding, we focused on potential biosignatures preserved in minerals. Minerals containing iron and sulfur, such as vivianite and greigite, are found alongside organic matter in less oxidized zones. This suggests that redox reactions that were caused by microbial life occurred. These iron-sulfur minerals could serve as potential biosignatures, preserving signs of past microbial life or biological activity on Mars. Here in the image to the left are fine-grained mineral nodules containing iron and sulfur, and on the right is a sketch that I created showing a close-up view of a rock cross-section showing a potential biosignature.
Now, to fully understand how these key findings were found, I will introduce three instrumentation and techniques to use in this experiment. To start off, we have the RIMFAX, which stands for Radar Imager for Mars Subsurface Experiment, which uses radio waves to imagine the subsurface rock layers which rovers drove across. The RIMFAX has collected the measurements and Bright Angel's foundation that helped establish the rock formation of its surface, which is highly important because scientists were able to discover the exact location of the mudstones and rocks on Mars' surface. Their positions and formation were determined thanks to the RIMFAX as well. In this study's objective, the geological history of rocks were identified with the RIMFAX with their chemical composition and conditions. And aside from the RIMFAX, the Pixel is another instrument used in the study, standing for the Planetary Instrument for X-ray Lithochemistry. The Pixel allows for the analysis of elemental composition of Martian rocks by measuring the elemental chemistry at sub-millimeter scales. The Pixel has allowed for the elemental analysis of mudstones that includes silicon, aluminum, and iron with some altered sediments, as well as the reactions found in the front cores that contain iron sulfides like greigite. And the Pixel is very important due to the fact that it is able to identify redox reactions found outside of Earth and allows for biosignatures to be confirmed. This study focuses on the possibility of rocks being able to contain and preserve chemical fingerprints from different time periods. Moving on to the third instrument presented, the Sherlock was paired with the Watson camera in this study, which allowed for the analysis of solid and rocks to determine past microbial life on Mars. Sherlock was able to identify and detect organic carbon G band in Bright Angel's vicinity based on the Raman signatures, as well as determining that strong organic signals correspond with less oxidized rocks. This is important because of the identification of biosignatures on Mars. This study sought signs of ancient life on Mars, which was taken with biosignature potential from the mission. And the image I provided included how Sherlock investigates a rock surface with the use of small circular lasers to determine chemicals, minerals, and matter in the surface that it's analyzing. And here I provided some images of the three instruments I talked about, provided by NASA to help you understand their structure and for visualization purposes.
Next, I'll talk about the techniques and instrumentation used in the study for the Mastcam Z and SuperCam instruments. Mastcam Z is a dual panoramic camera system mounted on the rover. Its powerful zoom capabilities capture high-resolution images of landscapes, outcrops, and geological features. They can even be color mapped to emphasize the oxidation gradients. So why is it important? Having the wide field, color, and stereo views give scientists the depth perception that is needed for understanding the spatial relationships to map the rover's path. In relation to the study's objective, Mastcam Z provides the context necessary for interpreting mineral and organic samples accurately. In the paper, for example, the image mosaic shows contact between the Bright Angel Formation and margin unit. Basically, Mastcam Z paints the big picture for scientists. SuperCam. SuperCam is a multi-technique remote instrument that analyzes rocks and soils from a distance with laser-induced breakdown spectroscopy. It can identify elemental composition of things like iron, magnesium, and calcium. It's important because it can remotely collect data and rapidly survey multiple outcrops. And in relation to the study's objective, SuperCam helped define mineralogical context of the outcrop and connected the chemistry of the nodules and reaction fronts to the surrounding environment. Overall, Mastcam Z and SuperCam showed where the rocks are, what they're made of, and how the redox reactions might have shaped them. And here's a sketch of the Mastcam Z, and as you can see, it has two cameras spaced apart like human eyes to create a 3D image.
And here are sources that we used, including some of the images and supplemental resources. And we just wanted to end off by saying that learning these concepts about redox reactions in this class is very relevant to current science research, because as we saw throughout this presentation, the findings of the study could potentially serve as evidence of past life on Mars, which could provide important evidence for future space exploration. Thank you so much.